So, hello everyone. So today, in this video, we'll study the we'll study the parts of the prokaryotic cell or the components of the prokaryotic cells. So, in last video, we have studied the definition of prokaryotes, that are single cell organisms, and the domain a three domain system of prokaryotic cell, and that is the domain archaea and domain bacteria. There are three domain system, that is domain eukarya, domain archaea and domain bacteria, but prokaryotes under the domain archaea and domain bacteria. Okay. Then we have studied the structure of prokaryotic cell or the components, which components are present in the prokaryotic cell, that is chromosome, ribosome, food granule, prokaryotic flagellum, plasmid, cytosol, plasma membrane, cell wall, capsule or slime layer. Okay, and we have also studied the plasmid. What is plasmid? The extra chromosomal DNA. Okay, that is made up of nucleic acid. Then we have studied the first component, that is cell wall. Uh, Basically, the outer membrane, the outer to the cell wall is called as the capsule. Okay, but I have started the from the cell wall here. Okay, we will study today the capsule, about the capsule. What is the structure of capsule and what is the function of capsule? Okay, we will study in this video. Okay, we have studied the cell wall and the gram staining technique. Okay, then we have studied the plasma membrane. Okay, the plasma membrane, thin lipid bilayer, that is phospholipid from archaea we have studied, and the phospholipid from bacteria. Okay, then then today we'll study about the capsule. Okay, so here, see, this is the what? This is a prokaryotic, a typical prokaryotic cell. Okay. Then there are these are the components, that is cell wall, cell membrane, flagellum, pilus, that is pili also. Here it is fimbria or fimbri. That is pilus is made up of a protein that is called pillin protein. Okay, and the fimbria or fimbri is also made up of a protein that is called fimbrine. Okay, pillin and fimbrine, these are two types of protein that are made up of this. Pilus is made up of a protein that is pillin, and fimbria is also made up of a protein that is fimbrine. Okay, pillin and fimbrine. Then this is the cell wall, this is the cell membrane, and here you can see. Okay, here, here you can see the capsule. Okay, capsule is the outermost layer of the typical prokaryotic cell, outer from the cell wall also. Okay, this is the outer membrane, that is called capsule. Then to the then to the inner, this is the cell wall, and this is the cell membrane. Got it? Then, okay. Then capsule is a large structure common to many bacteria made up of polysaccharides. Okay, we know polysaccharide, that is the classification of carbohydrate, that is monosaccharide, oligosaccharide, disaccharide and polysaccharide. Okay, polysaccharide is the what? Polymer of monosaccharides. More than two monosaccharides is composed to make polysaccharides. Okay. Then capsule is found in both gram negative and gram positive bacteria. Okay, we have studied what is gram negative and what is gram positive bacteria. We distinguished these two bacteria by applying the gram staining technique, right? We first what gram staining technique, you remember? Then first, yeah, first we took a cell plate. We don't know which bacteria, whatever bacteria it is. Okay, then we applied what? crystal violet, right? We applied crystal violet. Then we applied iodine, that is orange in color, that is iodine. Then we have applied the ethanol, right? Ethanol for decoloration. Okay. And why we took crystal violet, why we took iodine, why we took ethanol, I have mentioned all this in my previous video. Okay. So lastly, we lastly applied the what, safranin. Why safranin? Because to distinguish the gram negative bacteria. Okay, it directly attack to the what, decoloration bacteria. Okay, that is gram negative bacteria. So we have applied safranin. Then in this technique, in gram staining technique, we distinguish the both gram negative and gram positive bacteria. Got it? Then then see. Okay, most capsules are tightly packed. Okay, basically, capsules are tightly packed. When these are loosely arranged, then it is called slime layer. Okay, but there are exceptions also. So here most capsules are tightly packed. Okay, so they are difficult to stain. We can't stain it. Why? Most capsules are tightly packed. Okay, capsules are basically tightly packed. Then so they are difficult to stain. We can't stain directly. Okay. To visualize encapsulated bacteria, what is encapsulated bacteria? What is encapsulated bacteria? So, encapsulated bacteria is that which has no capsule. Okay, to visualize encapsulated bacteria using a microscope, a sample is treated with a dark stain such as India ink. Okay, what? India ink. So, why India ink? Why? Why in India ink? India ink can be performed easily. Okay. The capsule appears as a clear halo around the bacterium as the ink aren't penetrate the capsule. Okay. Okay. Here is encapsulated means what? Encapsulated means the bacteria. Here is the bacteria. Okay, when a bacteria is surrounded by a capsule, that is called encapsulated bacteria. Okay, this is a capsule, right? When a bacterial cell is surrounded by a capsule, a tightly packed capsule, it is called encapsulated bacteria. Okay. Then visualize encapsulated bacteria using a microscope, a sample is treated with a dark stain. Okay, we can't stain this type of bacteria uh, in a in a stain that is we used in for staining the gram positive and gram negative crystal violet. So with the help of crystal violet, we can't distinguish or we can't visualize. Okay. For visualizing this type of that encapsulated bacteria, we will apply the what, India ink. Okay, then see what is India ink. India ink can be performed easily. Capsule appears as a clear halo around the bacterium. See, under a microscope. Okay, this is a dark field, right? Dark field microscopy, light field microscopy. When dark field microscopy, the background is dark and we will apply the light to the sample. Okay, dark field, Dark field means what? Field means large area. So the large area, that is the background, is dark in color, and the sample is light. Okay. Then here, this is the encapsulated bacteria. Okay. By the help of India ink, we can see the encapsulated bacteria. The clear photo of what? Encapsulated bacteria. Got it? Then function of capsule. Okay. It serves as a shield, giving protection from toxins and from drying out. Okay. uh, this is a kind of what this is a kind of protection to this cell. Okay. How it protection from toxins and from drying out. Capsule is what capsule retain the maximum percentage of water. Okay. So it can it apply the water to the cell from preventing the drying condition. Okay. Then the capsule is considered a virulence factor because it enhances the ability of bacteria to cause diseases. Okay, capsule is considered a virulence factor. Why virulence factor? Yes, see, considered a virulence factor because it enhances the ability of bacteria to cause diseases. Why? How? So listen, first the capsule can protect engulfment of eukaryotic cells such as macrophages. Macrophages can't destroy the bacterial cell because the capsule is present. Okay, whether the capsule is present, this is a slippery like structure. Okay, it it can slip away. Okay, when the macrophage is going to engulf the cell, it can slip away from it, from the cell surface. So normally the macrophage can't engulf it. Okay, so that this is a virulence factor, it can enter to the cell. Okay. Then the capsule can protect cell from engulfment by eukaryotic cell. It also the, these are also the advanced the what these are also the advantages. Okay, that eukaryotic cell can't engulf the prokaryotic cell. Okay. Then capsules also help cells to adhere. Adhere means attach. Okay. then then here is some questions. We'll discuss the answers. Okay. First, which among the following is not a prokaryote? First, Saccharomyces. Saccharomyces cerevisiae is a what? Saccharomyces cerevisiae, the unicellular eukaryote. Got it? Then Mycobacterium is not a prokaryotic. This is eukaryotic. Okay. Mycobacterium is a prokaryote. Yes or no? Then Oscillatoria. This is also a prokaryote. Then nostoc. We know nostoc is also a prokaryote. So nostoc is also a prokaryotic cell. Then the from the which among the following is not not a prokaryote? Then Saccharomyces cerevisiae is not a prokaryote. Okay, it is a unicellular eukaryotic organism. Got it? Then what are archaea? Okay, what are archaea? Archaea are prokaryotes characterized as extremophiles that share some bacterial and some eukaryotic traits. Yes, this can be true. Okay, we have studied the three domain system, right? That is eukarya, bacteria, and archaea. The prokaryotes characterized as extremophiles, we know that. that share some bacterial gene, okay, gene which are similar in eukaryotic traits also. Then organisms that are adapted to high temperature environments such as volcanic springs. No, this is not right. This is not. Then single cell organisms that are killed by the application of antibiotics at certain concentrations. This is also not true. Then bacteria like organisms that can live only in extreme salt environments. No, this is also false. Primitive protist like creatures possessing fewer than two chromosomes per cell. No, this is not true. So, this is the correct answer. Okay. Then question, protists and bacteria are grouped into different domains because protist eats bacteria. No. Bacteria are not made up of cells. Huh? See, this is also made up of cells. Protists have a membrane bounded nucleus which bacteria cells lack. Okay, this this is true. But protists and bacteria are grouped into different domains because protists have a membrane bounded nucleus. Protist is an eukaryotic organism, whereas the bacteria cells lack because bacteria is a prokaryotic. Okay, then next, a component of prokaryotic cell. Okay, prokaryotic cell means these have cell wall. Yes or no? Cell wall is present, and we know plasma membrane is also present. DNA, that is the extra chromosomal DNA, is also present. Cytoplasm is the basic unit of life. The cytoplasm is also present. So the answer is all of the above. Okay. Then which of the following features is common to prokaryotes and many eukaryotes? Okay. See, let's see. Chromatin material present. Okay, then cell wall present. Features is common to prokaryotes and many eukaryotes. Cell wall present is present. Nuclear membrane present, common in prokaryotes. No, in prokaryotes nucleus is absent, so nuclear membrane is also absent. Membrane bound subcellular organelles. No, there is no organelle present in prokaryotic cell, so this is also false. So this is also false. Correct answer is the cell wall present in both prokaryote and eukaryotic cell, eukaryotic organism. Okay. Then gram negative bacteria take up gram strain. Gram negative bacteria take up gram strain. No, it's not true. This is false. Gram positive bacteria take up gram strain. Okay. Then which of the following feature can distinguish prokaryotes from eukaryotes? Presence of small vacuoles in prokaryotes, presence of cell wall in prokaryotes, presence of cell membrane in prokaryotes, presence of incipient nucleus in prokaryotes. How can we distinguish the prokaryotes from the eukaryotes? By the presence of incipient nucleus. Okay, incipient nucleus. But in eukaryote, the nucleus is well developed, right? Presence of cell wall prokaryote, then eukaryotes have also cell wall. Eukaryotes also have small vacuoles. Presence of cell membrane also present in eukaryotes. So we can't distinguish. But in presence of incipient nucleus is the feature of only the prokaryotes. Okay. So, what are plasmid made up of? I mentioned, we discussed about the plasmid, the extra chromosomal DNA. That is made up of what? Nucleic acid. Lipids? No, no lipid. Polysaccharides? No. Proteins? No. That is nucleic acid. Okay. the two domains to which prokaryotes are classified into. So we know the three domain classification. Yes or no? Domain eukarya, domain archaea, and domain bacteria. Yes or no? Domain archaea and domain bacteria is the feature of prokaryotic organism. Yes or no? So the answer is archaea and bacteria. Okay. When a water sample from a hot thermal vent was tested, it was found to contain a single celled organism. Okay, single celled organism. Okay, having a cell wall lacking a nucleus. What is its classification? Most likely. Okay, single cell lack of nucleus. Eukaryote? No. Eukaryotes have nucleus. Fungi? No. Protista? No. These are the eukaryotic organisms. Then archaea? Yes, the answer is archaea. Okay, because archaea are the extremophiles. These can live in the hot thermal vents. Okay, which of these is a characteristic of prokaryotic cell? Absence of cell organelles? Yes, there is no cell organelles present in prokaryotic cell. Okay, this is only the future feature of what? Eukaryotic cell. Absence of nucleus? Yes. Presence of 70s ribosome? We'll study about the 70s, the what 80s ribosome in the organelle that we will study in the ribosome part. Now we have studied only the cell wall, then cell membrane, then capsule. Okay. After that we will study the ribosome, the cytoplasm. Okay. The and eukaryotic all cell organelles we will study. Okay. So the answer is all of these. Okay. What is the shape of a bacterial plasmid? Okay. We have studied bacterial plasmid. Linear? No. Circular? Yes. Irregular? No. Bacillus? No. Bacillus means, bacillus means rod shape. Okay. Rod shape. No, that is not. So, what is shape of bacterial plasmid? It is circular double-stranded DNA. Got it? Circular double-stranded DNA. Okay. Okay. This is the last. Okay. Bye. Thank you.